Tomato Biotechnology by Javier Namagata and Sonali Moinani. Genetic transformation is a change caused by genes and involves the insertion of one or more genes into an organism in order to change the organism's traits. Genes in organisms are turned on and off through a process called gene regulation. When a gene is turned off, it no longer provides the directions for making proteins. This means that the proteins needed to fulfill a particular job aren't produced. To turn off a gene, there are a series of steps that must happen during protein synthesis. The complementary strand of DNA or antisense strand is complementary to the sense strand. The two DNA strands form a double helix. The two RNA strands produced from these DNA strands are also complementary. When bound into a double helix, the RNA strands cannot function to produce proteins, which results in the extermination of a gene. Modern technology provides us with the ability to genetically engineer plants, such as tomatoes, using the new techniques of gene isolation and insertion. These techniques allow us to construct transgenic plants that contain and express a single, well-defined gene from any source. This enables us to improve plants, such as a tomato, in various ways. Tomatoes have various undesirable characteristics, yet thanks to modern technology, people are able to genetically modify and improve these characteristics. Tomatoes contain pectin, which are polysaccharides that help bind cells together in the cell wall of a plant. An enzyme in tomatoes called polygalacturonase lowers the amount of pectin tomatoes contain, which causes the tomatoes to turn soft during ripening. The softness of the fruit increases the possibility of it inheriting fungal infections, which damages the fruit. To prevent this from happening, scientists genetically modify tomatoes. To fix this problem, polygalacturonase synthesis in tomatoes is turned off. To do this, an antisense gene, which is a single-stranded RNA that is complementary to an mRNA strand, is created. When mRNA forms a duplex with a complementary antisense RNA sequence, translation is blocked. This intervenes with the manufacturing of polygalacturonase, which results in more developed and flavorful tomatoes. Ethylene is used to treat fresh market tomatoes to give them their red color. However, when ethylene is synthesized at a certain point in fruit development, it marks the final ripening. In the genetically engineered varieties, ethylene production is blocked with antisense techniques so tomatoes can be picked later, at a more flavorful stage, and do not over-ripen and rot. Once picked, they can be ripened when commercially appropriate by simple exposure to external ethylene. Several genetic engineering strategies are available to protect crops from viruses. One of the strategies is the coat protein, or CP strategy. In CP-mediated resistance, the gene responsible for the outer protective wrapper of the virus is introduced into plant cells. The presence in plant cells of this viral gene prevents the virus from efficiently making copies of itself, thus reducing the damage of the tomato. This is similar to what we did in the Piglo lab. In the in-class experiment, we transferred plasmid DNA into bacteria to make it fluorescent green. The gene that was taken from the jellyfish and was genetically transferred into bacteria was a transgene. In both experiments, we added new DNA to get the cell to produce new proteins. Just like we transfer the GFP gene into the bacteria to make it glow, farmers transfer the coat protein, for example, into tomatoes to make them resistant to viruses. Biotechnology will have a significant impact on all our lives and future generations. Like any tool, biotechnology is being used to solve some of our problems and provide benefits to various fields of science. With biotechnology, we can improve selected attributes of microbes, plants, or animals for human use by making precise genetic changes that were not possible with traditional methods. Biotechnology has already been used to enhance the ability of plants to fight disease and pests. Many new crops are under development that will have enhanced nutritional content. It will also be possible to remove undesirable substances from food, such as allergies. The enhancement of tomatoes is just one of the examples of the impressive results of biotechnology. Thank you for watching!